So I think, as typically we've seen with cancer vaccines, some of the limitations include finding a relevant tumor antigen. Not only a tumor antigen that's relevant to a specific tumor type, but obviously we see different antigens among different patients and even sometimes within the same patient. The other issue is generating an effective immune response in patients who are heavily pretreated. And so I think with these 3 vaccine platforms that have been discussed, the first, again, was using a high-fidelity fusion oncogene. The second abstract with SYNC-T was using essentially cryolysis to develop an in situ vaccine. And then the third is using an oral vaccination method. And so these are different types of platforms and perhaps may be able to overcome some of the issues with the tumor microenvironment that typically restrict the infiltration of immune cells. But together, these abstracts also, I think, raise an important question of should we be using therapeutic cancer vaccines to generate an immune response first? And of those patients who are immune responders, is that the time that we follow with immunotherapy? And it doesn't necessarily have to just be standard, immune checkpoint inhibitors, but we have a wide variety of immunotherapeutic agents that perhaps can better address T cell exhaustion, which again is typically one of the reasons sometimes why these vaccines do not work.